Hello, everyone, and welcome to today's live broadcast, Cannabis Testing Making Life Easier in the Laboratory, presented by Kathy Stenerson, a principal scientist in workflow development at MilliporeSigma, the life science business of Merck KGaA in Darmstadt, Germany. I am Kaylee Bach of LabRoots, and I will be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is the leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the Ask a Question box located on the far left of your screen. Type in your question and hit send. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, please click on the Help Desk button located in the promotional board at the bottom center of your screen, or use the Ask a Question box to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the Continuing Education Credits tab located in the top right corner of the presentation window and follow the process to obtain your credits. Please join me in welcoming Kathy Stenerson. I will now turn the presentation over to her.
Thank you very much, Kaylee, and thank you everyone for logging in to watch this today. What I'm going to be talking about are some approaches for those of you that are interested, or maybe you're already doing testing of cannabis in an analytical laboratory. Some of the approaches that we've found to help some of the pain points for some of the tests that are done. So what we're going to cover, I'll start with pesticide testing, which is a required test in many states in which medical and recreational cannabis is legal. Then I'll talk a little bit about potency testing, a couple of new approaches for doing residual solvent and testing for terpenes, and finally I'll wrap up with mycotoxins. So doing pesticide testing. On flower material, you start basically with a beautiful bud, and you have to go through a series of manipulations in the laboratory to get an extract that you can then inject into your analytical instrument. And in the case of pesticides, many of the required pesticides that laboratories have to test for require the use of both GC and LC mass spec to be able to cover the entire list. Also, too, some of the issues when doing this type of test, the extracts can be very, very messy. As you can see from the picture in the upper right-hand corner, they can be green, they can have other things in them that interfere with detecting the pesticides of interest. So that's an issue, is being able to clean that extract so it can be injected in the instrument. Also, in the case of doing LCMS, these extracts are inorganic solvent. So injecting that directly into an LCMS, depending on your starting mobile phase conditions, the peak shapes of the first few peaks can be very poor, and I'm going to show you a way to address that. So let's start with the extraction. Many labs, when they extract pesticide residues from cannabis, use an approach called Koechers, and that is an acronym, which stands for quick, easy, cheap, effective, rugged, and safe. It was originally developed for doing pesticide residues out of fruits and vegetables. It's a very simple technique, doesn't require much equipment. It simply uses loose salts and sorbents, organic solvent, and a centrifuge and a tube. So the salts, what they do is sample is homogenized, it's put in the tube, then an organic solvent, usually acetonitrile, is added, and the sample is shaken to extract the pesticides. At that point, then, salts are added, and what these salts do is they cause any water in the sample to separate from the organic solvent, and the pesticides will be in the organic solvent part. Then the organic part is taken off, put into another tube with loose sorbents that act as a cleanup. So once again, the sample is shaken, and then the unwanted background sticks to the loose sorbents. There are two official methods that describe this technique. One is in the AOAC, and the other is a European method, and they're listed on the right-hand side of the slide. So as far as the salts go, what kind of salts are used? Well, there are different salts depending on what it is you want to do. When the technique was first developed, they started out by just using sodium chloride and magnesium sulfate. And then as the technique became more sophisticated and applied to more and more pesticides, they started coming out with different salt combinations to offer in buffer and capacity. And once again, the salts act as partitioning agents, and they optimize the extraction efficiency. So they also help to drive the pesticides into that organic layer. Now, as far as the cleanup goes, the cleanup sorbents, which are loose also, there's 3 very common ones that are used and available from multiple vendors. PSA, which stands for primary, secondary amine, and this takes out things that will interact with amine groups, so fatty acids, organic acids, polar pigments, sugars, things like that. Carbon, which takes out colored interferences, mainly chlorophyll and carotenoids, and C18, which by hydrophobic interaction will take out fatty background. These are very common sorbents. Now, there's a new class of sorbents made with zirconia, and they're available from MilliporeSigma called Z-Sep sorbents. And these have some unique characteristics. They remove lipids, pigments, and they can be combined with other sorbents to provide even a wider array of cleanup. So these well, actually the bar should go down to the C18, but these top 3 sorbents, they're described in the official methods, the AOAC and the European methods for doing this Koechers extraction and cleanup. And as I said before, the Z-Sep sorbents are unique. So they're a new type of sorbent that can be used for this Koechers cleanup. And remember that the role of the sorbents is to trap the interferences that co-extract with the analytes, that come out in the acetonitrile layer with the analytes. But at the same time, we do not want those sorbents to remove our target analytes, which are the pesticides. So let's talk about that for a minute. Okay, so when using a conventional mixture that is very common in Koechers, PSA, C18, and carbon, which we show in the graph as PSA, C18, NV-carb, that's our version of carbon. What's shown here is recovery of pesticides that were spiked into cannabis and then extracted back out with the cleanup. And after the cleanup, the recovery of the pesticides using this PSA, C18, NV-carb mix are shown by the purple bars on the chart. And most of them are not bad, okay? They're above 80%, 70%, which is considered acceptable for this type of application. But there's a problem with one, this canoxyphen here. Now, there is an alternative mix that can be used for doing a cleanup in Koechers, and it's called Sapelq Verde. It's a mix available from us. And it utilizes one of these zirconia sorbents to help trap interferences. As far as recovery goes, after cleanup, the Verde offers some advantage. Overall, it offers higher recovery, 75% versus 71%. And as far as reproducibility, it's very, very comparable to using the conventional mix. Its main advantage, though, is that it will not reduce the recovery of compounds that would stick to a carbon, the NV-carb that's in the conventional mix. And canoxyphen, which is the structure is shown here, the structure of canoxyphen is planar, okay? So it will stick to these graphitized carbons quite strongly. Now, the Verde mix has a specialty carbon in it that is not as retentive for these planar molecules. So hence, we get a much better recovery of this canoxyphen pesticide using the Verde mix compared to the conventional mix. So moving to the LC side of things, and I mentioned that the pesticides have to be run by both GC and LC because you can't do all of them by one technique or the other very well, okay? So the Koechers extracts are inorganic solvent, and the LC methods start out with a lot of times there are gradient methods, and they start out with high aqueous to get pesticides to retain on the column. So there's a couple issues with that. First of all, when injecting an organic solvent into this mixture, your early eluting peaks tend to not look very nice. They're kind of ugly, they're kind of broad. Another thing is that during that Koechers extraction, in addition to those pigments and those lipids and things and sugars that can come out along with the pesticides in the extraction, cannabinoids come out too. So obviously you're doing cannabis, you're going to have cannabinoids. They're going to co-extract with the pesticides. And the levels are quite high. So these cannabinoids will not be removed entirely by any cleanup, unfortunately. So what happens is when you inject them into an instrument, they end up going into the detector. Now with GC, it's not as much of an issue because they go through the column, and they go through the detector, and then they go out to waste. But in LC, they can go into the detector, and they can get the detector quite dirty. That requires that you take it apart more frequently and clean it. So one approach for doing that would be to try to adjust your chromatography so that the cannabinoids that are still left in that extract with the pesticides will elute later on the column or after the pesticides. The idea being is after your last pesticide goes into the detector, if you have a valving system between the detector and the column, you can switch it to waste, so the cannabinoids can then dump to waste. And that's what we investigated here. Now, most people, when they're doing HPLC, they reach for a C18 column. This is very, very common, okay? And what we did here was we show a comparison between a typical pesticide run, and these pesticides, many of them are on the list required by the state of Oregon. And we've overlaid them with the same run conditions with the major cannabinoids that are present in cannabis. And you can see that they, in red, that's where the cannabinoids are, they elute pretty much in the middle of the pesticide. So there's no way you're going to be able to do that dump step that I talked about, because if you try to dump the cannabinoids to waste, you're going to dump some of your pesticides too. But if you choose a different column, and in this case we did the same experiment but with a column called the RP amide, we found that the pesticides were eluted a little bit earlier relative to the major cannabinoids. So in that case, we could possibly take advantage of a dump step to dump those cannabinoids to waste. And we can still use the amide to analyze our pesticides. So once again, cannabinoids are co-extracted with the pesticides. They can contaminate the LCMS. So what we're doing here is trying to elute them later than the pesticides so we can dump them to waste. Now, in addition to column choice, the gradient that is used can also help. Using, in this experiment, what we did was we compared using acetonitrile in our gradient to methanol to see if we could get the cannabinoids and the pesticides to elute even further apart. So the top chromatogram, we're using a methanol gradient, and the black is the pesticides and the red is the cannabinoids. Now on the bottom, we're using acetonitrile gradient. The black is the pesticides, the red is the cannabinoids. And we found that we got a little bit better separation between the cannabinoids and the pesticides when using acetonitrile. So combining an amide column with an acetonitrile gradient gave us the best separation of all. So we were able to use that dump step to dump the major cannabinoids to waste before they enter the detector and not lose our analytes of interest. So now on to the second issue when injecting these extracts into LC-MS systems, especially when you're using a gradient. You're starting with a high organic, you're injecting, a high amount of aqueous in the mobile phase, you're injecting organic into it. What can happen? Early eluting peaks will tend to broaden out because you have solvent mismatch with the mobile phase, and that's what's happening up here. With our amide column, the first amide column that we looked at was an amide column that was made from a particle called a fused core. And this particle has a solid center and a porous shell. And the idea behind it is that it's a very highly efficient particle so that you can run separations highly efficiently but faster. Okay, so that's the best way to go if you can. But what we found was when we switched to an amide made from a fully porous particle, that's one that does not have that solid core, we got a little bit better peak shape, most likely due to the fact that the fully porous particle could accommodate this slug of organic a little bit better, and also we got better mixing. Now, by attaching a guard column to the front of that fully porous column, we improved the peak shape even more. So we are still able to inject that organic slug into the starting mobile phase conditions and get halfway decent peak shapes for our early eluting peaks. What some labs will do to deal with this ugly peak shape issue is they'll take their Koechers extract and dilute it into aqueous so that they can inject it and get decent peak shapes. But if you dilute your sample, of course you're going to raise your reporting limits and also create an extra step in your workflow. So by just using a fully porous column with a guard, you're creating a little extra mixing and improving your peak shapes of the earlier eluters. And using a guard column is a really good idea anyway when you're running these really messy extracts. It's going to increase the life of your more expensive analytical column. So putting all this together using our amide column with an acetonitrile-based gradient, a fully porous particle structure in the amide column, and a guard, these were the results we got when we were looking at cannabis that was spiked with pesticides. What the TIC on the top shows is the pesticide elution pattern. Pyridibin was our last eluting pesticide. It came off at 13.3 minutes. And the chromatogram right below it, I pulled out the major ion for cannabidiol and THC, two major cannabinoids that you see in cannabis, to show where they elute relative to the last pesticide. And you can see that they come off after. And for other major cannabinoids that are present in cannabis too, on the amide column we know that they come off even later than THC, so you should be able to dump those to waste as well. So what about the GC side of things? Because I mentioned you have to run those extracts by LC and GC. Well, in GC, a couple of the interfering classes of compounds are terpenes and cannabinoids. Unfortunately, nothing is going to preferentially retain terpenes, so you just have to deal with that. Maybe choose the right ions to look at. If you're doing MSMS, which you probably are, the right transitions to look at where you don't get interference. As for the co-extracted cannabinoids, they do come through the column, and Sapelcu Verde mixture will reduce these a bit, the acidic ones specifically. So we do find that we get a total area counts at looking at background. We do get a slight reduction over using a conventional mixture, 35% reduction in background versus 31. So just staying on GC for a minute, maintenance is really important, especially when you're doing these types of samples. Because repeated injections of a green extract, even after cleanup, it's light green, but it's still a little green, you can end up with contamination in your liner. And I show a couple examples of different GC liners with residues in them. After 10, 20 injections, sometimes you'll get these residues, and these residues create active sites where pesticides can end up degrading and absorbing, and so that your responses will be reduced. Also, if you have a seal at the bottom of your inlet, that can get dirty too. And that's what I show here is a brand new seal, which is nice gold color, compared to two other ones above it. The one on the right has been exposed to injection of agricultural samples extracted by Koechers, and the one on the left is just age. So these need to be replaced and periodically. So moving from pesticides into potency testing, this is probably the #1 test that is done because we need to know what the content of the major cannabinoids are in the products that we're selling to consumers. Quantitative analysis is extremely important. Processing can affect cannabinoid content and consistency, especially for extracts, because many times when extracts are made from concentrates that have gone through some kind of process to convert all the acidic forms into neutral. And in the case of THC, you probably already know the neutral form is the form that's psychoactive, or I should say psychotropic. Now here in this application, what we did was we looked at a couple different forms of edibles, gummy bears and brownies, and we used a very simple Koechers-like extraction followed by a UV analysis for looking at these, HPLC UV. Okay, so we used HPLC for our analysis, so why not GC? What's the difference anyway? Let's take a look at a GCMS analysis of THC. Nice peak, retention time of 22.7 minutes. And that's the mass spectra of it. Now let's take a look at a GCMS analysis of the acidic form. Nice peak, similar retention time, that's interesting. And similar mass spectrum. Okay, so what happened here? Well, it's pretty well known that acidic cannabinoids can be converted to neutral cannabinoids by heating them. They decarboxylate. And in the case of THC, that's essentially what happened here. It decarboxylated when we injected it into the GC because the injection port is hot. And what we're detecting is the neutral form. So if you were trying to do a sample that had both the acidic and neutral forms in there, you couldn't distinguish them. They would just elute as one peak as the neutral form. HPLC, on the other hand, can distinguish them. There's no heat involved. So what I'm showing here are delta 8 and delta 9 THC, the neutral forms, and then the acidic form. So they're they're distinct peaks, well resolved. So that's why HPLC is the method of choice for analyzing for potency. Now, these edibles that we took a look at, the first one was a brownie, and we looked at chocolate brownie with frosting. And for the extraction, we did a very simple Koechers-like extraction. So similar to what we did with the flour material when we were looking at pesticides. So we weighed brownie, put it into a tube, added acetonitrile to it and shook it up, and then added salts, shook it, and centrifuged it. And there was some fat that came out of the brownie along with the cannabinoids that we were looking for, co-extracted out. So to do the cleanup, we put the organic layer in the refrigerator for a few hours, and the fats just precipitated out. It was pretty simple, and it took a few hours, but you could go off and do something else during that time. And then took that extract and analyzed it by HPLC. And for the spike level that we spiked at, which was 40 ppm, micrograms per gram, the recoveries were pretty good. They were all above 80%, and our reproducibility for spiked replicates was very good with RSDs below 5. Now, for doing residual solvent testing and terpene testing, you could use headspace for residual solvents, and a lot of labs do a solvent extraction followed by GC for terpenes. So that's two different approaches, two different sets of equipment. Or a new approach would be to use a technique called solid phase micro extraction, or for short, SPME, and you could use that technique for both. Now, what is SPME? If you're not familiar with it, that's a whole nother course, but I can just briefly tell you how it works. Essentially, it's a micro extraction device, and it consists of a fiber, a coated fiber, which is shown the center picture there. And that coated fiber is exposed to samples. A lot of times it's used for headspace sampling, but it can also be immersed in aqueous-based samples. And the analytes then will be extracted by the fiber. The fiber then is taken out of the sample, brought over to a GC, and put into the injection port and desorbed directly by the heat of the injection port. So this illustration explains the steps that are involved with doing an extraction. In the first step, you put your sample, first of all, in a vial that had some kind of septum cap. The SPME device, and what's shown here, is a manual device. So the actual fiber that does the extracting is inside of the little needle that's sticking down from the holder. We call that the needle assembly. So you use that needle assembly to puncture the septum in the sample. Then there is a plunger on the top of the holder that you depress, and that exposes the fiber inside of the sample. After the extraction is complete, you bring the fiber back up inside that needle assembly, then take the whole thing out of the sample, take it over to the GC, and reverse the process, and expose the fiber when you're inside the GC inlet. And by thermal energy, the analytes are desorbed off. They go onto the GC column, and you just do your analysis just like normal. The advantage of SPME is there's no solvent at all. It's solventless. So you don't have any organic waste, and also the fiber is reusable. So unlike a solid phase extraction where you use the device once and throw it away, you can use this device over and over and over again. And what I've shown here is how it's being used manually. It can also be automated very, very easily. There's vendors out there that make autosamplers that can accommodate SPME, and I'm just showing the one that we have in our lab that it has a magnetized head on it, so it can pick up the sample, move it over to a heated agitator, put it in the agitator, and it can actually do the sampling. And what's shown here is it's actually inserting the SPME fiber assembly down into a sample, and then it pushes down on the plunger and exposes the fiber. And then after it's done, it goes over and goes into the injection port of the GC and desorbs. So you can just set the samples up and walk away. It's really very nice. And these are called XYZ autosamplers, and they can also be used for other things too, liquid injection and other things too. So they're multi-purpose, and they can be used for SPME. So I used SPME first to look at residual solvents. And since I am in the state of Pennsylvania, medical cannabis was just made legal recently, and we don't have access to concentrates and whatnot. And at the time that I did this work, I used hemp extract. And I spiked it with a list of residual solvents that were of interest to those that are doing this type of testing. And I extracted it by SPME and then quantitated it against a curve that I prepared in soybean oil and extracted by SPME. Now, why did I prepare a curve in soybean oil? I have to explain one thing about SPME that sometimes is people don't really understand off the bat, but unlike SPE, solid phase extraction, which is an exhaustive extraction, SPME is what we call an equilibrium extraction. So it doesn't extract everything out of the sample. So therefore, you do an extracted curve in order to quantitate samples. Not a big deal, especially if you have automation. You just set up the curve and you run it, and then you run your samples afterwards. And that's what I did here. I made up a curve in soybean oil and then ran my hemp oil right after that I spiked, and I also ran some hemp oil unspiked. Now, you'll probably notice that butane and isobutane aren't on my list, and I know that those are common solvents that are looked for labs doing this type of testing. And the only reason is that they would work just fine by the method I'm going to show you, but I just didn't have the right kind of setup to spike those gases into my materials accurately. The column that I chose is a porous layer open tubular column. It's a Pellew plot, and I chose it because if you do use butane and isobutane, this column will retain them and separate them from all the other analytes of interest. And I use GCMs in full scan mode. Okay, so these are my calibration curves out of my soybean oil, and I got pretty good linearity, as you can see here. I was actually getting such good sensitivity with this method that at about 70 ppm, I was starting to see a little bit of overload on some of the compounds. And the cutoff, I know for Oregon, it varies depending on the solvent you're looking for. But generally, if you can see down to 10 ppm, that's going to be fine for most of these, which SPME has no problem with. It's an enrichment technique, very, very sensitive. So in my spike temp oil, I spiked it at 10 ppm. And for spiked replicates, my recoveries were above 80% for all the compounds, and my RSDs were very good. I got very good reproducibility. They were below 10%. Now, hexane is missing from my bar chart here. That is because I detected hexane in my unspiked hemp extract at 59 parts per million. So obviously I know what they use to make that extract with. Okay, terpenes. Now this technique, solid phase microextraction, SPME, can also be used for doing terpenes, either qualitative profiling or quantitative analysis. Now in this application here, we had some cannabis that we had obtained through NIDA, and we did a terpene profile on the material, just on dry flower material, by exposing an SPME fiber to the headspace. There's different types of SPME fibers available, and just the one that you select depends on what you want to do. So I chose one that would be very good for getting low molecular weight and very volatile compounds. So I chose the one that's described here, this DVB carboxin PDMS. It was a very, very simple extraction. I just exposed the fiber to the headspace over the dry plant material for 20 minutes after I had warmed the sample up for 30 minutes to get it equilibrated. And I was able to identify 45 different terpenes in that plant material. Now I did that using a combination of GCMs and retention indices. Also, too, SPME can be used in a field sampling mode, and these samplers can be taken out in the field. They're available in little sealed holders, so you do your sampling in the field, and when you bring the fiber back inside of the assembly, it's sealed off with a septum, and then they can be shipped back to the lab and analyzed. And so we did a something called the Cannabis Boot Camp out in Portland, Oregon, a couple years ago at Chalice Farms, which is a dispensary there, and we used these SPME field samplers to look at terpene profiles from 3 different varieties of cannabis that they took out of, that they had there at their facility, and that's me on the far right, and I'm sampling one type, and I had 2 helpers that were sampling 2 other types. And the picture below, the second picture, just shows that all we did was just expose the SPME field samplers to the cannabis flowers for about 5 minutes. Then we shipped the samplers back to MilliporeSigma in Bellefonte, Pennsylvania, and about 2 weeks later, I desorbed them into a GCMS system. This was the first variety that we sampled, Hippie Crippler, the second variety, Slimer, and the third variety, which was Super Orange Crush. And you can see we got very nice terpene profiles, and we were able to see some differences between them. So this could be a pretty useful tool if, say, you wanted to do some terpene sampling for qualitative reasons, perhaps out in the fields, for identification of a strain or other reasons. Now, quantitation of terpenes using SPME. We also worked up a quantitative method, and this method had some different parameters than our qualitative method. And we chose to look at 3 specific terpenes that are of interest, pinene, limonene, and linalool. And we studied them from the cannabis variety that we had in-house that we had obtained from NIDA. We spiked, what we found was when we did the qualitative determination, we then analyzed our cannabis quantitatively, and we found that the terpene content on it was very, very, very low. Okay, we don't know how old the cannabis was, we don't know what variety it was. So in order to make it closer to the varieties that are being sold in dispensaries now, we had to spike the cannabis with known levels of terpenes to bring it up to relevant levels. and that's what we did. So we spiked our cannabis and then quantitated it against a curve that we had prepared and extracted by SPME. And then we compared that to that spiked cannabis sample that we prepared by a conventional extraction method. Many labs use an ethanol extraction, which is what I'm showing on this slide, to produce an extract that they just shoot into a GCFID to look for terpenes. The extract is a beautiful green color, as you can see, and it also turns the GC liner a nice green color after about 10 runs we found. So what we did was we took this spiked extract, we extracted it with the ethanol method I'm showing there, and we also extracted it using our Headspace SPME method. And what we found was that the results were very close to each other. Reproducibility was good for both techniques, and the accuracy was good for both techniques. The difference is that with SPME, we did not have to use any organic solvent. It was also a hands-off technique. We set up the samples and walked away. There were basically 2 steps, just putting the cannabis in the vial. We found adding a little water helps, putting it up on the instrument and walking away. Whereas, as you can see with the ethanol extraction, there's essentially 3 to 4 steps that are required, and you still end up with a green extract that kind of mucks up your instrument. So why GCMS for terpenes? Here's another thing, too. A lot of labs use FID. GCMS is better. Why? Well, if you use FID, you're not going to be able to know for sure what you're looking at is what you think it is. And what do I mean by that? Well, let's take a look at this peak at 19.81 minutes of a terpene peak by GCMS. Nice peak. If we look at the spectrum on the back half of it, compared to the front, there are some similar ions, but you can see that they're very different. The reason is these are 2 terpenes that are colluting with each other. Looking at that peak, you would never know, okay? But if by looking at spectra on the front versus the back, you can tell that they're essentially two things coming off together. So what I did was I subtracted the back from the front, and I was able to identify it as this compound here that I'm showing on the bottom left. So GCMS helps to identify for sure these peaks as specific terpenes. Now, is spectral searching, like I just showed you, is that sufficient by itself to identify terpenes? Not entirely. For example, let's say we have 2 terpene peaks, one at 25.85 minutes and one that eludes at 25.97 minutes. Let's look at their spectra. This is my first peak at 25.85 minutes, and it is alpha langine, I think that's pronounced. Now, the second peak is identified as a completely different terpene, yet they have very similar mass spectra, very similar, almost the same. So if you tried to identify those peaks by spectral searching, you could potentially misidentify them. So in this case, it's good to know the retention times that you're looking for. So that makes having standards very important. Okay, retention indices is a little more complicated to use. That's a nice tool as well, but the main thing is the standard. So if you're looking for a specific list of terpenes, you want to have standards of known composition so that you know the retention times of those terpenes, and also you want to be able to make sure that you're not having anything colluting with a terpene. So using standards combined with spectral identification, you're going to get correct results most of the time. So the last topic I'm going to talk about is mycotoxin testing, and this is a realm of testing that's not quite as common as the other things that I've talked about, but more and more we're starting to see that states are requiring this. I do believe that California is going to require this. Now, mycotoxins are produced by fungi that are naturally present on the cannabis plants. The thing is, though, is that mycotoxins are toxic to humans, and some are suspected of causing cancer. So they're analyzed by GC and HPLC, mainly HPLC, after some type of extraction and cleanup. And mycotoxins are commonly analyzed in grain, and a common cleanup for this type of analysis involves something called an immunoaffinity column. But these columns have some issues associated with them. They're expensive. They have to be stored under refrigeration. There is another way, though, to do the cleanup and using solid phase extraction. And we have a cartridge that's called the Spectral, and there's a series of them for doing mycotoxin cleanup. And so we tested one of these cartridges out with a cannabis extract to see how well it would clean it. So we extracted the cannabis as if we were looking for mycotoxins. It was a beautiful green color, as you can see there. Then afterwards, we spiked it with mycotoxins, and we ran it through a cleanup cartridge called the Supervisor. It's a simple pass-through. And then we looked at the recovery of the mycotoxins and also looked at the resulting extract to see how clean it was. So after passing it through the cartridge, you can see it went from this emerald green color to a nice clear, maybe pale yellowish color. So it was fairly clean. And there was no precipitate in the final extract either. Now, as far as the mycotoxins we spiked in, this was the LCMS-MS analysis that we did for recovery on those. And the recoveries were good. So we didn't lose them on the cleanup, which is very important. So a cleanup is if it cleans something really well, that's great. But if it takes away your analytes, you really can't use it. So that's everything that I have for you today, and I want to thank you very much for your attention and just let you know that we're dedicated to making your life in the lab successful. So don't hesitate to contact us if you have any questions or if you need help with anything that you're working on. So thank you very much, and at this point I'll turn it over back to you, Kaylee.
Thank you, Kathy, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question drop-down box located on the far left of your presentation window. Type your question into the box that appears on your screen and click the Send button. We'll answer as many questions as we have time for. Let's get started. Our first question is, I'm doing Koechers extraction on cannabis for pesticide testing and use PSA C18GCB for the cleanup. Can I substitute the Verde mix directly, or do I have to change my method?
Actually, you can just substitute it directly. You don't have to do any other types of changes to your method. It was designed to be a direct substitute. It will offer the same advantages of PSA C18GCB, so it will remove the sugars, it will take out pigments, it will take out fats. The added advantages, we have a specially engineered carbon in that mixture so that it will not reduce the recoveries of the planar pesticides nearly as much as GCB would. So yes, you can just directly substitute it.
All right. And this next question is a two-part question. It says, will other cannabinoids besides THC convert to neutral form if analyzed by GC? Is there any way to use GC and still distinguish neutral and acidic THC from each other?
Other cannabinoids, if they're acidic, they will also convert. So yes, you're going to see them only as the neutral forms. If GC is all you have and you want to try to distinguish the two forms, one approach, and it's a little more complicated than HPLC, is to derivatize the sample. So you would essentially be derivatizing the acidic group on the acidic forms of the cannabinoid so that you could get a distinct peak with a separate retention time from the neutral form. There's some drawbacks to derivatization. It's an extra step and whatnot, and so a lot of people shy away from it, but you can do it.
All right, and we have time for one more question. It says, what equipment is required to do SPME?
To do SPME, to get started with it, all you basically need is a holder. Like what I showed in one of my slides, I showed a manual holder and a set of fibers. That's pretty much it. And the vials, most labs already have that. So if you just wanted to try it, you could just start with that. And it's also advantageous to have a hot plate to warm the sample on. So those three things are enough to get most people started, and a ring stand too. There's accessories that are available that especially to hold things that make life a little easier. And then eventually, if you find that you like the technique and it's working for what you want to do, many labs will then convert an automated system like what you saw on my slides. I have an XYZ auto sampler that has a head on it for SPME, and all that equipment's all readily available. So technically, you don't have to do any changes to your GC just to try it. You don't need a special inlet or anything. You just need to have a holder and fibers.
Wonderful. Thank you again, Kathy. Do you have any final comments for our audience?
I just want to thank the audience again for tuning in for the webinar today, and look forward to hopefully hearing from some of you.
Great. And I would like to once again thank Kathy Stenerson for her presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through June 29th, 2018. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.